Sponsored by Department of Biotechnology, CTEP New Delhi, Ministry of Science and Technology, Government of India. In this, I extend my hearty welcome to you all. It's the time to invite dignitaries onto the dais. I deem it a great privilege to invite the propelling force behind this lecture series, the man of exemplary and transformative leadership qualities who thrive to kindle scientific temperament in the students, that none other than our principal, Dr. Ramchandra R.K. sir, onto the dais. Roshni honored the sir with sapling. I feel proud to invite Regional Joint Director, Commissionerate of Collegiate Education, Zone 2, Dr. C.H. Krishna sir, whose remarkable achievements and leadership serves as an inspiration to all, onto the dais. Pawan Sri honors sir with the sapling. I am glad to invite a distinguishable, capitative individual as a chief guest, Dr. P. Vijay Nirmala Madam, Head Zoology, Principal, University College of Science and Technology, Adikavi Nannaya University, Rajamahendravaram, onto the dais. Nandini honor Madam with the sapling. I consider it a great honor to invite energetic, erudite, regal, genius, and the man of simplicity, Professor Y. Suresh sir, Faculty, Department of Animal Biology, University of Hyderabad, to grace the dais. Rishita honors sir with the sapling. I would like to invite capitative and supportive Head of Department of Biotechnology, Government College, Rajahmundry, onto the dais. Jyoti honor Madam with sapling. I invite the person of dedication towards duty, Dr. A. Annapurna Madam, IQAC Coordinator, to grace the dais. I invite Academic Coordinator, Dr. Sanjeev sir, to grace the dais. Sai Lakshmi honors sir with a sapling. So now the stage is set. We go for lighting the lamp. I request the dignitaries to lighting the lamp. I invite faculty members also to please come on to the dais. I request students to be please keep quiet. I invite Sushma for prayer song.
शुभं करोति कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपो ज्योतिर्परब्रह्म दीपो ज्योतिर्जनार्दनः दीपो हरतु मे पापं दीपज्योतिर्नमोऽस्तुते.
Thank you, Sushma. Now the stage is set. I handed over the program to President of the lecture series. Thank you.
So I request Program Coordinator Dr. Anusha, please come and occupy the chair. And request one of the student honor her with a bouquet. So very good morning, ladies and gentlemen, and today's chief guest, Regional Joint Director of Zone 1 and 2, Dr. C.H. Krishna Garu, and Principal of College of Science and Technology, Adikavi Nannaya University, Professor Vijay Nirmal Garu, and the man behind this today's program, Professor Y. Suresh Garu from University of Hyderabad, Telangana, and the head of the Department of Biotechnology, Dr. Nageshwari Madam, and academic coordinator of the college, Dr. Sanjeev Kumar, and the program coordinator, Dr. K. Anusha, and other members of the college, faculty, and dear students. I am very happy to be part of this wonderful program. So, it's the first time in the history of the college we got funding for lecture series, popular lecture series from Department of Biotechnology, Ministry of Science and Technology, Government of India. So I really appreciate and thanks to the Department of Biotechnology for enhancing the quality parameters in the Department of Biotechnology, and also I really extend my thanks to the Biotechnology Department of our own college, Nageshwari Madam and Anusha, who taken care for planning meticulously to execute this particular program. And the Department of Biotechnology, Government of India is coming forward to offer this kind of popular science lecture in relating with biotechnology and advances in Biotech Chronicles, that is the title. And it is everybody's responsibility to understand to upgrade our knowledge in relating with biotechnology. See, the biotechnology which leads two powerful fields on this globe. The first one is health and the second one is agriculture. So health and agriculture are the prime sector for human beings. In both these, the biotechnology and innovative technology which leading us. So one of the advanced techniques which we are using to understand ourselves means our body. So that means the biology which combines with computer science, we call it as a bioinformatics, and which is the tool to understand the mechanism of DNA. And if you understand your DNA and the secret code of each and every human being on this blue planet, if you understand it, and then you can live years means hundreds plus years. So that is exactly, it can be the computational understanding of DNA is possible with bioinformatics. So that means each and everybody has its own DNA, and if you know the genome engineering, so that we can avoid many diseases. So nowadays, which we are protecting ourselves by taking just general medicines, by testing our samples and getting our medicines. But it is not the right process to root out the particular diseases in our body. That is possible if you understand your DNA and the secret codes behind that particular DNA, then we can go for gene-based medicines. So that is a front-end technology for the human being that is possible through bioinformatics and biology. Not only that, and target-oriented drug delivery is also one of the fact. And also, the biotechnology it has its own applications. That means diversified fields. So, like health and also agriculture and environment also. So, as we know all, the COVID-19, so we understand that particular virus, and we are happy to feel ourselves, being Indians, we are the persons to develop a vaccine to this world. So that is possible with this bioinformatics and biotechnology. So that is the importance of this particular field. So that's why the government of India has come forward. Previously we know that only DST, CSR, like this funding agency. But the importance of health and agriculture sector, these are the two main key areas for human beings for this globe. So that's the reason the government of India came forward to offer extempore research in the field of biotechnology. That's why a separate biotechnology division is there. That's the funding and startups and seminars and then projects, identifying the star colleges. These are all the schemes proposed under that particular scheme. So happy we receive the financial assistance to conduct a wonderful event of popular science lectures. So the information, the knowledge, what we are learning this particular 3 days, today, tomorrow and day after tomorrow. So that is also these 3 days very auspicious for us also, because we planned this year a week-long National Science Day celebrations, that is the 23rd to 28th. Tomorrow also we have college open day. So these are all there. So really we feel it is a science week, and we are happy to conduct this kind of lecture series with eminent personalities like Suresh Garu from Central University, and another tomorrow's speaker is also coming from the same Central University. So that is also a very happy thing. That means we have been associating while contacting, while calling to our college, and then we are going to establish a strong, academic understanding to Central University Hyderabad to us, because yesterday I spoke with sir, and then he has given assurance of that. So this second week of March, so they are offering 20 internship for 20 students with his laboratory and everything, the facilities and everything taken care by them. So that is also a very happy thing, that in the second internship we are going to have. And not only that, he is the man to give an assurance that to go for other funding opportunities across DBT and DST, or also like that. So this is very happy occasion to share all the things, and I once again congratulate the Department of Biotechnology for conducting this wonderful lecture series in this college. Thank you very much. So I request head of the department, and also I invite our IQSC coordinator Sri Annapurna Garu onto the stage, and also I request Biotechnology head of the department Dr. B. Nageshwari madam, please give away his initial remarks of this program.
A very happy morning to you all. Myself, Dr. B. Nageshwari, Head Department of Biotechnology and an organizing secretary of this program now, the popular lecture series on advances in Biotech Chronicles. Myself, along with organizing committee, cordially welcome the esteemed dignitaries on the dais and off the dais, and also all the delegates. Sir, even though being from a non-science background, life sciences background, he has talked wonderfully. I think we should give a big clap to sir for understanding the life sciences part also, that DNA thing and all. It's mind-boggling. Okay, so as we all realize, we all are in a situation where we are delivering our services in our own jobs on one side and regularly updating our knowledge on another side. And despite this, of course, our fields of teaching and research are not optional. In fact, I feel that we are most essential for the society and thus for the progress of the nation. Teaching and research are the key factors linking to the quality of people's lives. Last few years have been seen to have invested a very significant time and effort in the area of research, especially during the COVID-19 situation. So this has created clear opportunities and areas of larger networking. And now we all are a part of that large network. So this program with the theme Advances in Biotech Chronicles is the right platform to bring various such stakeholders under one roof to discuss the developments and update our knowledge among ourselves and younger generation. And also, the talks in this program are designed to drive the students, researchers and teachers to pursue research and take it to next higher level. So on this occasion, I would like to take the opportunity to thank our principal, Dr. K. Ramachandra Rao Garu, for encouraging and supporting us in every way in getting this program right from sanctioning to implementation. Next, I thank the guest of honor and the chief guest for gracing the event in spite of their busy schedule. Next, I thank Professor Y. Suresh Garu from Department of Animal Biology of University of Hyderabad for readily accepting to deliver the talk. I hope the stay and arrangements are comfortable, sir. We are trying our level best to make it comfortable to you. And then about Dr. K. Anusha, one of the speaker of this event, I would like to share that she has joined the Department of Biotechnology in August 2023 on general transfer, and she was awarded PhD in December 2023. And I'm proud to say that she has proven to be a good asset for the department. I thank her for working hard for this event. I also thank Parnika, a very young lecturer from our department, and the students for their incessant contribution to the department. And I thank the DBT CTEP under Ministry of Science and Technology of Government of India, which has accepted our proposal to popularize biotech activities through popular lectures. Finally, I thank each and every one of you who are contributing to the success of the conference, and we are looking forward to your kind attention throughout the program as we are trying our best to ensure that your time engaged in this program to be fruitful and you all go back with rich information. And also feel free to give your feedback for us to improve and serve you better. With this, I convey my best wishes to each and everyone here. Happy learning. Thank you all.
I request today's chief guest, so Dr. Sechi Krishna Garu, Regional Joint Director and a proud alumni of this college. So we are always remember you as alumni rather than RJD sir. So I request him to give his message on this occasion.
Good morning to all. Dignitaries on the dais, Principal of this college, Professor Ramachandra, and President of this function, and today's guest of honor, Professor Vijay Nirmala, Principal Science College, Adhika Nanna University, Rajamhendravaram, Professor Suresh Garu, University of Hyderabad, Telangana, Dr. Nageswari, HOD Biotech, Dr. Anusha, Program Coordinator, Dr. Annapurna Garu, IKC Coordinator, Dr. Sanjeev, Academic Coordinator, and faculty members, students, research scholars, persons from print and electronic media, ladies and gentlemen, a warm greetings to all. At the outset, let me congratulate the Department of Biotechnology of Government College Autonomous Rajahmundry for organizing CTEP program of DBT. I also extend my thankfulness for inviting me at this event. Being a geologist, I am aware of advancements in the geological field, and I regularly update myself by discussing with my colleagues and friends. But biotechnology is not my basket to explore with. In order to promote biotechnology innovators in India, the Government of India, Department of Biotechnology offers financial assistance for the arrangement of conferences, seminars, symposia, workshops. Additionally, researchers receive travel support to present their papers at conferences held abroad. The DBT also provides assistance for setting up DBT stalls at exhibitions both nationally and internationally. Financial support is further extended for the organization of popular lecture series. Collectively, these 4 activities are referred as CTEP, representing conference, travel, exhibition, and popular lectures. Congratulations to the Department of Biotechnology for getting selected for this program. Over the past few decades, biotechnology has emerged as powerful force, bringing about unprecedented breakouts that have transformed the landscapes of science and medicine. Let us embark on a journey through the key milestones that mark the advance in biotech chronicles. Firstly, the decoding of human genome stands as monumental achievement in biotechnology. The Human Genome Project, completed in the early 2000s, provides us with the entire sequence of DNA that make up the blueprint of human life. In the realm of healthcare, biotechnology has unleashed a wave of innovative therapies. Precision medicine, gene therapy, and immunotherapy have become game changer in the fight against various diseases. Biotechnology has also revolutionized agriculture, addressing the global challenges of feeding a growing population. Genetically modified crops have been developed to enhance resistance to pests, diseases, and environmental stress, ensuring and biotechnology has further accelerated process. Machine learning algorithms are being empowered to analyze vast biological data sets, leading to faster drug discovery, more accurate diagnosis, and a deeper understanding of complex biological processes. However, with these incredible advancements come ethical considerations. The power to manipulate life is its most fundamental level raises questions about the responsible use of biotechnology. Let me list some of the remarkable achievements in the biotechnology field. Vaccine development, genome sequencing, bioinformatics and computational biology, stem cell research, crop biotechnology, biopharmaceuticals, medicinal biotechnology, environmental biotechnology. The advances in Biotech Chronicles are a testament to our collective commitment to unlocking the mysteries of life and harnessing its potential for the betterment of humanity. As we continue this journey, let us remain vigilant, ensuring that the ethical dimensions of biological progress and the benefits are shared equally across the global community. Together, let us embrace the promise of biotechnology to create a healthier, more sustainable and interconnected world. I once again congratulate the organizers for arranging this wonderful program and also thank them for inviting me this program. Thank you. Jai Hind.
So RJD sir has a preoccupied schedule with him. So he asked me to lead the occasion. So before that, it is a token of gratitude and love of our proud alumni and regional joint director of Zone 1 and 2. So we are going to honor him with shawl and memento. So I request the participants, please. Dear students, so front line లోపలికి వచ్చేయండి. Okay, please. Now I request Principal of Science and Technology, College of Science and Technology, Adikavi Nane University, Professor Vijay Nirmal Garu will speak on this occasion.
A very good morning to one and all. Respected President and Co-Principal, Professor Ramchandra Garu, today's guest of honor and speaker, Professor Y. Suresh Garu, Head of the Department, Dr. Nageshwari Garu, Coordinator of this program, Dr. Anusha,
So please, dear students, please settle down.
IQAC Coordinator, Dr. Annapurna Garu, Academic Coordinator, Dr. Sanjeev Garu, faculty colleagues, scholars, other invitees and dear students. Indeed, it's a great pleasure for me to be a part of this program. Before me, RJD sir has briefed you some of the importance of this biotech. I congratulate the team for getting funding for this wonderful topic, that is advances in biotech chronicles. Indeed, students, it's the time that you all open your eyes and see the happenings that is being carried out by the biotechnological field. The synthetic biology has opened the doors to fix the problems of the genetics even before the baby is out of the womb. Detecting the diseases using biopsy was a common, but liquid biopsy has made it easier to detect the diseases. And if you go to the coding has opened the boundaries in the field of medicine, agriculture, food science. In agriculture, CRISPR precision has enabled the development of genetic manipulation, where we are able to even get the taste of non-veg in the vegetarians also. Moreover, the technology, that is AI and artificial intelligence and machine learning, has added on along with IoT. So using IoT, we are able to detect with the help of sensors the health status and the personalized case to case. We can track the health of the individuals also. So as bioinformatics was already briefed by the principal, I just want to add the bioinformatic languages are made easy for the data analysis and also to detect the diseases. The regenerative medicine, that the stem cell therapy, even it has given life to the dead cells. So this is a big change which has brought in the life of the individuals. The bioprinting. So we only knew about the X-ray. The bioprinting, 3D printing technology has made a way to create living tissues, organ transplantations easy on personalized tissue and organ transplantation. And digibiomics. So the digibiomics, that is the analytical diseases, outbreaks are being detected. That is how it was possible for the case with COVID-19. And cell therapy. So if you come to the cell therapy, it is really, so suppose the own people they married, there are some diseases which are already known. And by knowing that there is some disease, already the baby is carrying some defective genes, it is fixed with the help of this cell therapy and stem cell technology, where with the help of some stem cell technology, we have designed even the cancer cells can be treated also. So pharmacology, so the different fields in which biotechnology has been penetrating into, though it's life sciences, it is having interrelation with pharmacology, and the immuno-oncology, where we can detect the cancer cells at the initial stage, and we can even modify the T cells and develop the immunity, and the vaccines, development of the vaccines for the cancer cells is also possible. The biomanufacturing has paved the way to synthesize the living organisms, bioenzymes, biofuels, biopetrol, which are naturally very expensive, and by using synthetic methods, we can come out of this problem, which is of high demand. The nanobiotechnology is another wonderful field which is used to, and there are different ways how we can deliver the drugs exactly to the affected cells and lessening or reducing the impact on the healthy cells. The neuroscience and neuroinformatics, especially this is used in treating the neurological diseases, and for example, if you take the neuro-HIV AIDS, the brain is, they're trying to modify the cells in the brain. And food technology, and no doubt the biologicals. And finally, if you come, we can even talk about the biocosmetics. So here are many girls, Nordic girls, boys are also very interested in this biocosmetics, where eco-friendly cosmetics are produced which can give more care to the skin naturally. And so students, this wonderful lecture is being organized for you with the efforts of the Department of Biotechnology and the principal. So it's a great opportunity for you to grab the opportunity and interact with the eminent personality, that is Professor Suresh sir. Utilize this platform to open the ideas that you have within you and work hard and move forward, and maybe in future you will be one of them to innovate something better which is no one has discovered yet. Wishing all the best, and I'm congratulating once again the Department of Biotechnology, the principal and all the staff. I thank you for making me a part of this program. Thank you one and all.
Hello. Thank you, madam, for enlightening front-end technology of biosciences. Now, all biotechnology students are there here. So I would like to ask one question. Dr. Jennifer Daudna, anybody knows it? Dr. Jennifer Daudna. He's a famous biotechnology entrepreneur. He's the man to discover a gene editing tool that is CRISPR-Cas9, CRISPR, CRISPR-Cas9. So that is the tool. By using that, he can edit the gene and understand the secret codes in it, and with respect to that, he is going to advise medicine. So that is a very popular entrepreneurship which is undertaken on the platform of biotechnology. So in USA, that is, he's been practicing billions of rupees business. This is a health sector, how the biotechnology diversified. So we can understand. So that is one of the, I just asked Google, who is the biotechnology entrepreneur? That is Google given me Dr. Jennifer Daudna. He's the man who invented a gene editing tool by using computational technologies. So that means to understand the DNA and its secret codes, and by chasing that, he's going to suggest a therapeutics. Yeah. So that is gene therapeutics. So that's a big sensational news in biotechnology. So that means I got doubt how the biotechnology technologists turn into entrepreneurs. So that means this is the way they are doing it. So small enterprises, okay, that is okay, but a big entrepreneurship initiated with this particular technology, gene code editing technology. So that's what I want to share. And then our IKSC madam, all the best. Our academic coordinator, please.
Thank you, sir. So, right, so మన ఈ బయోటెక్నాలజీ అంటే ఏంటండి ఇంతకి? What does it do? So in any field, technology is integration of machinery and devices. I mean, it deals with...
From one of my alma mater, that is Hyderabad Central University, I deem it to be a privilege to introduce Professor Suresh with a very brief biodata, because sir has many adorations with respect to academics. So Professor Suresh sir, today's speaker, is a faculty in the Department of Animal Biology at University of Hyderabad. He pursued his master degree in biochemistry from Sri Venkateshwara University, Tirupati, then completed PhD in the Department of Biochemistry from Usmania University, Hyderabad. Sir has remarkable academic qualifications and research experience. His areas of research are reproductive biology, anti-infectious agents, reproductive toxicology, and oncogenesis. Sir is an editor for several reputed journals. He is also a recipient of several awards. To name a few: Professor G.P. Talwar Middle Career Scientist Award, Professor S.S. Guraya Memorial Gold Medal and Oration 2019, Bharat Vikas Award 2018, and many others. Sir has mentored 8 PhDs till now, and 5 are ongoing. Sir has altogether 78 publications in reputed journals, and he has conducted 30 programs like conferences, symposia, workshops, invited lectures, etc. He has been principal investigator for 12 projects under various funding agencies like DST, CSIR, DBT, UGC, ICMR, SERB, and Institute of Excellence, University of Hyderabad. With this brief biodata, I welcome Sir to deliver his speech.
Good morning, all of you. How many are graduate students here? How many are graduate students? I mean BSc.
UG students, raise your hands.
Okay. Okay. How many are PG students? Most? Only few. I hope all of you are from biology department, right? I am trying to be comfortable with you because I am little tensed because we don't see such a huge class. Our classes are around 30 people, not more than that. So seeing so many people, I am also tense. So we will try to get to know each other first. Any PhD students here? No. PG students? Few of them are here. How many of you want to do PhD after PG? PG students, anybody here? I think the front row is PG students, right? Only two. Who else are PG students here? Raise your hands. PG students, I mean postgraduate students. Okay, very few of you are here. Fine. How many of you want to do PhD after MSc? You don't want to do? Okay, how many of you want to become teachers? How many of you want to become teachers? I am also a teacher. None of you? You don't want to become teachers? If you don't become teachers, who is going to teach the next generation? Right? Pardon me? You can have the mic and talk to me. I am trying to get a rapport with you before we actually talk about the presentation, which I am sure will be very boring for you, but I will try to impress as much as possible. Okay, at the outset, I should thank Anusha madam. Actually, I told her, madam, please excuse me, I cannot come, but she called me so many times and she made me to come here, and I am very, very happy to be here to see all of you. And I should also thank Ramchandra sir, very dynamic sir. Though he is physics, he talks a lot of biology. I understood it yesterday and today, and I am sure this college will flourish under his guidance. Thank you very much. Okay. I asked a question, how many of you want to become teachers? None of, only one or two raised their hands. Okay, I ask another question for you. Who is more important or who is better, the teacher or the mother? Raise your hands if it is mother. Teacher or mother? I'll go for teacher. What is the reason? A mother can put one baby to sleep, whereas a teacher can put the whole class to sleep. Right now, tell me who is better? The teacher. You have to accept it. That's the reality, right? Who has the power to put all of them to sleep in a whole class? Is the power of the teacher. So you should become teachers so that you can make people to, when we are in the USA or somewhere, okay, it's time to sleep, let's go for a lecture. That's how we used to do. So nicely go and sit in a air conditioned room and have your sleep and come out of it, right? And later on have coffee or whatever. Okay. So I hope the projector is working. Oh, it's here. I have two lectures here. One is very basic one. The other one is primarily my research one. Go to second. Since there is no projector here, I will talk from here. I hope it's fine with you, right? Okay, I made 2 lectures. Let me talk to you about our research work. Okay. First, let me try to impress you. There is a very interesting work we have done over the last 4 or 5 years. Okay. So, how many of you are from agriculture background? I thought Rajahmundry is a 2-tier city and many people are agriculture. None of them are raising. How many of your parents are into agriculture? Right? So many people will be from farming sector. So when you want more crop production, what do? You add these fertilizers. Fertilizer is one for the growth of the plant. The other one is to protect the plant. What do you add? Pesticides. Students, right? Pesticides you add to protect the plant. Not only the growth is important, but also you have to protect the plants from pesticides. That's very, very important. Okay. So not only pesticides, we use many compounds in our daily life. How many of you use mosquito repellents? For mosquitoes, we put that coils, right? Or we put the vaporizers, right? These are all a lot of chemicals that we use every day. Okay. And we are encountering chemicals knowingly or unknowingly every day through different sources. Okay. And other question I'll ask is, when they switch on the presentation, you should notice for one small point, right? ikkade unchandi paraledu, no problem. I can speak here. Please. Huh? So do your teachers use PowerPoints for presentations? Yes? Okay, I am also using the same. But whenever there is a lecture, they switch on the PowerPoint, and before this, okay, I will put it this mode. You have to observe one point here. What is that? Look at the left side bottom corner. Slide 1 of 79. Okay, you can decide this fellow is going to talk for about 79 minutes. And it is time for me to relax. Okay, so any presentation, that is what I do. Whenever somebody puts up a presentation, the first thing I see is how many slides this person has. Okay, basing on that, I will decide, okay, he is going to talk for long time or short time or whatever. Okay, I am trying to make you little comfortable. Okay, we will come to our research work, what we have done. This was an excellent work which was published in very good impact journals, around 8.9, 10 journals. We are looking at the effect of specific compounds which we call as the pyrethroids. And this study is very important because we have conducted the studies basing on what humans are exposed. This is the first study in the world that we have done. Everybody does studies with some concentration. They give the chemical to the animal or chemical to the human beings and see the effects. But this time we have exactly calculated how much of the dose the humans are exposed. Similarly, same dose was used to conduct the experiments. Okay, you can have a look at it here. I will give a brief introduction. As I said, our agriculture output has come up very nicely in the last 40-50 years. If you look at from 1961 to 2020, the agriculture output has come up. The reasons being access to more land, more water facilities, and more development in the technologies. Okay, there are many reasons, but at the same time, the food production also has increased. Not only the agriculture, along with the agriculture, the food production also has increased over the period of time. It's very good because there was a revolution. Professor M.S. Swaminathan was the person behind it to increase the food grains. as the food production increased, there was also a parallel increase in the amount of chemical pesticides that's being used in the country. So one of the reasons why the food production increased is people started using lot of pesticides. It's not that they have increased the agricultural land, because every crop is known to be affected by pests. The amount of pesticides that are increased is very high, starting from, it's in the tons, 10,000 tons to almost 80,000 tons in the country. Before we go there, we should define what is a pesticide. Pesticide is a substance or a mix of substances intended for preventing, destroying, repelling or mitigating a pest. As you can see here, they spray the pesticides on the crops. Okay. So these are the chemicals which will kill the pests. Pest means any kind of pest. It can be insects or bacteria or fungus or even animals. So when you use a lot of pesticides, what happens is there's a lot, it prevents pest outbreak. The crops are not damaged by the pests. That's one advantage you have. The second advantage is instant benefit. Nowadays people are interested in organic farming. I hope you know what is organic farming. Organic farming is the one where you don't use any pesticides, but the yields are very less. The yield of the crop is very less if you go for organic farming. But if you go for pesticides, the benefits are very good. The crop is protected and you get a lot of financial benefits. And potential yield. Potential yield means you have more yield of the food product. And finally, the quality of foods at affordable prices. See, if you go to a market, supermarket, there will be rice of two brands. One is the regular rice, the Sona Masira whatsoever, and there is something called as organic rice. So there will be definitely a 3 to 4 times price difference in the regular rice and the organic rice. So you have affordable prices. If you use pesticides and have more of the crop, the prices are affordable. So that's the reason why pesticides have been used. Well, advantages are always there. It also comes with disadvantages. Using of pesticides results in, they are very harmful for health. I hope you know that. And threat to biodiversity. Suppose I have a crop and I want to have a good yield. Naturally there will be pests, there will be insects that will be coming. If I spray these pesticides, all the insects that are supposed to exist, nature has produced them, they are supposed to, but we kill them. Using pesticides also kills a lot of butterflies. Nowadays you don't see butterflies on the crops. The reason why we don't see butterflies on the crops is because they spray a lot of pesticides. Most of them have been eliminated. You don't see many, only in forests or somewhere where there are no pesticides, you see the butterflies and biodiversity. And economic drainage. Economic drainage means you spend a lot of money. You know, you have seen in newspapers that farmers spend a lot of money on the fertilizers, and this causes a lot of economic drainage. If you go for organic crops, you don't have to spend any money there, but sometimes they have losses also. Okay, and the final one is the contamination of soil and water. You add pesticides. If you go to Kerala, where I have seen them, here at least in Andhra Pradesh and Telangana, they spray the pesticides. In Kerala, they dissolve the pesticides in the well. They throw the pesticides in the well and pump it. Just imagine, if pesticides are dissolved in the well and watered, it goes down, plant takes up, along with water it takes up pesticides also. So this is how water, and the same water is being used by people, groundwater, the pesticide goes into the ground and it gets contaminated. So use of pesticides has both advantages as well as disadvantages. So we can't help it. Right, coming to our country, India is the fourth largest producer of pesticides in the world. We export a lot of pesticides. Of course, China and USA are the first and second. If, among the so many countries, India is the largest producer of pesticides. And the Indian pesticide market is about 42,000 crores in the year 2020. So you can imagine how much pesticides are being produced and sold. 42,000 crores is not a small amount. And among them, insecticides dominate the Indian crop protection market. So we use more of the insecticides. Pesticides are different types. Insecticides, rodenticides, fungicides, bactericides. So, but insecticides are the most commonly used. So the total as well as per hectare consumption of pesticides in India shows a significant increase after 2009 to 10. So for every acre, earlier people used to go only for organic farming, but later on pesticides have been introduced and the use per acre has gradually increased. This is the trade data. I am just giving an example so that you can appreciate how much India is earning money by exporting a lot of pesticides. If you look at the trade data, this is the exports and imports of pesticides by the Indian market. As I was telling, pesticides are divided into many of the classes: herbicides, fungicides, insecticides, rodenticides, and fumigants. You can see the classification there. Mostly India uses insecticides, and they are again divided into two types. One is natural, the other one is synthetic. Among the natural, you have plant-based and mineral oils. These are the two types which people have developed. And among the synthetic, there are organic and inorganic. And coming to organic, again, there's a lot of classifications: organochlorines, organophosphates, carbamates. I have put pyrethroids in red because these are the compounds which we use for our research work, and non-corticoids. So this is one of the review we published earlier in a very good journal, that is Reviews in Toxicology. What is the projected pesticide use in 20? That was, I am talking 3 years back. This is the projected pesticide use in the year 2020 globally. As you can see here, insecticides, they constitute about 4%, herbicides, and these are other ones. And of course, if you look at the graph here, the down, the pesticides consumption in different states, different countries. Of course, India uses very less amount when compared to China and United States and Argentina, but we are still using less amount of pesticides, but still it is a lot of pesticides we are using. Coming to Indian scenario, you can see what are the types of pesticides that are used. The plant growth regulators are constitute a lot of the bulk, and you also have fungicides, and you have insecticides, right? So the graph B, again we published this earlier, shows what are different types of pesticides people use. You have something called as fenvalerate, xylofruthine, alethrine, deltamethrin, and cypermethrin. So cypermethrin is one of the pesticides that is very commonly used. Okay, right. So the other graph shows the uses of pesticides in different states of the country. You can see the highest consumption is by Maharashtra, and of course we are lucky that Andhra Pradesh still is in the last place. Of course we also use, I don't know why Andhra Pradesh uses less pesticides, but Maharashtra uses, reasons I don't know. But coming to this one class of pesticides which we call as pyrethrins and pyrethroids, as you can see here, initially they are derived from a flower known as the Chrysanthemum cinearifolium. You know chrysanthemum, right? What is it called in Telugu? Chamanti. Mudda chamanti. I know it is chamanti. Okay, right. So they dry these flowers and they derive this specific compound known as the pyrethrins, and later on they also synthesize something as synthetic pyrethrin. These are the structures. I am just showing you what are the different types of pyrethroids. Pyrethrin 1, 2, cynarin, cynarin 2, jasmolin. Jasmolin is again from a flower, jasmonia. Okay, so pyrethroids are one of the serious compounds among the pesticides. Where do we use them? Number one, agriculture. Commonly used in agriculture to protect rice, wheat, cotton, sugarcane, groundnut, sunflower, and other crops. And in domestic purpose, that's what I was telling you. We use this pyrethroids in domestic purpose in the form of mosquito coils, mosquito nets. There are also mosquito nets which come coated with pyrethroids, and in animal houses, public gardens. You know, when you have seen the municipal office people coming with the spray, right, fogging. That fogging has a lot of pyrethroids, especially alethrine to kill the mosquitoes. And how do we take it into our body? I am telling you, everybody is consuming pesticide every day. There is no doubt about it. Okay, we all have to realize that we consume pesticides every day. How? Through food, oral ingestion. Through food, as I said, pesticides are sprayed on the crops. They are all dissolved in the water. They get into the food we consume them through the oral route. The second one is use of mosquito coils. That is, when you use mosquito coil, we inhale them. Don't think that putting that vaporizer is good for you. One side you are driving away the mosquitoes, other side you are inviting health problems. So don't use them. There are a lot of natural compounds that you can use to keep the mosquitoes away. Okay, because this is very important for you, right? You are the new generation. I am telling you, I am going to show you some data which will be very interesting for you, especially for new generations. And occupational exposure. Many people know, if you look at farmers, they mix the pesticides with hands. They don't wear gloves, so they get exposed to a lot of pesticides, right? Right. So coming to these compounds, I am showing a little bit cell biology here. These pyrethroids, they are very, how do they act on the insects? So if you see there are two figures here, it's a normal condition. In a normal condition, these pyrethroids acts on the nerve membranes. You know what are nerves, right? What are nerves? I am not giving a lecture, I am taking a class here. So in our classes, we keep asking a lot of questions. Right? What are nerves? Anybody? They are the cellular components of the nervous system. Right? The brain has nerves, the sympathetic and parasympathetic has nerves. Right? So these pyrethroids, they act on specific cells known as the nerve cells. On the left panel, you can see that it is a normal condition. The membrane potential is normal, the sodium current is normal, action potential is normal. But when you add pyrethroids, what happens is they go and bind to the proteins, which we call as the receptors on the nerve membrane. Nerve has a membrane and it binds to them. And of course, what happens is you can see the change in the graphs. The sodium current is changed. Both type 1 and type 2 pyrethroids, they cause this. And the action potential, you know what is action potential in nerve? I am sure. Action potential. You know it, right? So that's also disturbed. So when you add these pyrethroids on the insects, it acts on the nerve cells and disturbs the action potential, and the insect becomes aggressive, hyperexcited, and it causes whole body tumors. The insect will have tumors and then, tremors, whole body tremors. And of course, the same thing also happens, seizures can also happen, and that's how the insects are killed. Have you ever seen when you light a mosquito coil, what happens to the mosquito? You never experimented it? Mosquito will not die, it will just fall. If you remove after some time, it will again get up and go away. Okay? So once it falls, what you should do? Nowadays you get bats, right? Mosquito bats. That's more easier to control, but still, you have to chase them and then kill them. Right? You have to be behind mosquitoes. You can't do that over the whole night, right? How many use bats here? Maybe 1 or 2 hours you will do that. After that you have to sleep, right? You cannot be doing it for the whole. Okay? So, as you can see here, what are the different health problems that you get because of this pyrethroids? It's very alarming, very dangerous. Look here, pyrethroids, they cause carcinogenicity. Continuous use of pyrethroids in the form of mosquito coils or vaporizers, or if they are sprayed on the pest, there are a lot of cancers that have been reported. They also cause epigenetic changes. What are epigenetic changes? Epigenetic changes are changes those occur during the course of life, like the DNA gets modified, methylation, acetylation. I hope I am not saying too much here, but these epigenetic changes are also caused by these pyrethroids. Okay, and neurotoxicity, as I said earlier, that's also if you, I don't know how many of you have really, looked into this. The reason why I got into this was, when I moved to Hyderabad in 2007 from US, I rented a house and there were too many mosquitoes. So we used to light coils. Fine, mosquitoes are gone, but I could not sleep. Any time we light a coil, I was not able to sleep. Then I was thinking, what is happening here? The day I don't light the coil, I sleep well. But the day I light the coil, then I was reading and I found out that these coils have alatrin, is a chemical which will act on your nervous system and make your nerves very hyperactive. That's why you become more active and you don't sleep. Okay, it causes neurotoxicity. So that, and it also causes a lot of respiratory distress. It has a lot of effect on the respiration. Immunotoxicity, people who are exposed to these pyrethroids, they have very weak immune system. Their immune responses are not good. And the area we are interested was reproductive toxicity. I am basically a reproductive biologist, where we work on the effects of different proteins on sperm maturation and fertilization. So this is our area, and of course people have earlier shown that it also has reproductive toxicity. So pesticide contamination is there in the food, you can read it. There are a lot of documents. Indian Council of Agriculture Research has provided the residual levels of different pesticides, including synthetic pyrethroids in major food commodities. Anything you buy in the market has pesticides. Let it be a fruit or a vegetable or rice, wheat, milk, even your Pepsi and Coke and Sprite. Why should they have Pepsi, Coke? And, there's a lot of controversy on Pepsi, Coke and Sprite, maybe 10, 15 years back. The reason is the water they use for preparing these beverages are derived from the groundwater and they have to be decontaminated. So they add these pyrethroids into the water, decontaminate it, of course at very low concentrations, and you cannot remove that. Once you add it, it is dissolved. There is no way of removing the pyrethroids from the water that is used for preparing your so-called fashion drinks like Pepsi, Coke, Sprite. So there also you get it. Anything, even your bottled water like Bisleri, any bottled water will have pyrethroids. Okay, so there is no way you have to drink, then there is no way. Okay, so Indian Council of Agriculture Research, they estimated, okay, and they have come up with values. And the other one is, there is another organization, the ICMR, Indian Council of Medical Research, of course National Institute of Nutrition, which is the institute located in Hyderabad. They estimated that an average consumption of different food items, only rice and vegetables, and reported that 388 grams of food is consumed by Indian every day. We consume 388 grams of rice and vegetables. Reason why I am saying is we have correlated this with the doses we have used in the animal experiments. Okay, here I am showing you how much pyrethroids we are taking every day. I am talking about only pyrethroids. This is the amount of pyrethroids we are taking every day. This is a fact, you have to believe it. Cypermethrin, right? The amount of food consumed per gram, it is 789, okay, and total level of pyrethroids, okay, it is 598. So totally, if you look at, there are 1, 2, 3, 4, 5 pyrethroids which are generally present in the food, and you are consuming 941.746 micrograms per day, every day. This is reality. Okay, so this is provided by ICMR as well as by Indian Council of Agriculture Research. This is the amount of pyrethroids we are taking. Okay, through food, only through food. So basing on that, we designed our experiments. I am talking more about research here. We use rats as the model system. Okay, and there is a lot of relation between rat and humans. Why, people ask, when you want to test something for humans, why are you testing in the rats? The reason is the physiology of the rat and the humans are almost same. Okay, that is the reason why we test it. And 1 day of rat is equal to 26.7 human days. So that's why rat doesn't live for long. So if you look like that, and 13.8 days rat days is equal to 1 human year. So if rat is alive for 14 days around, so it has lived the life of 1 year of a human being. Okay, right. So that's what you can look here. If rat age is 1 month, it is equal to 2.5 years of humans. If it is 15 months, it's equal to 37.5 years of humans, right? So basing on this, we came up with some rationale, the reason why we have to do it. The health hazards of pyrethroids they are studied at high doses. Everybody has used high doses. Doses used in such studies do not reflect human settings. So whenever this whatever I am telling you, whenever you are planning a research study, ensure that it applies to the human settings. There is no point simply doing some animal experimentation. And studies on the effects of pyrethroids at doses that are equivalent to human consumption were never reported. So this is the first study we have conducted, and it has received a lot of appreciation. It was published in newspapers also. Okay, so we had these different objectives. So what we did was, basing on whatever data we had, I hope this may not be confusing for you. So we have calculated how much of each of this chemical that should be given per it doesn't work. Okay, so you can see here, basing on the body weight of the humans and how much food they are consumed, we have decided that we will give 9.97 micrograms of cypermethrin, 1.5 micrograms of deltamethrin, 0.538 micrograms of phenvalerate, 3.35 micrograms of xylothrine, and 0.323 micrograms. Total it will be 15.687. That is actually equal to human consumption. So we have planned these experiments. We had two types of exposures, exposure 1 and exposure 2. So in exposure 1, what we have is the animals were exposed to doses that is 1/5 or 1/10 of the actual amount of pyrethroid that are consumed by the humans. And EP2, we call it exposure protocol 2. Here we have treated the animals with exactly the amount of pyrethroids that we consume every day. Okay, then we treated them for 9 to 15 months, and of course in the other one we have treated them for 1 to 15 months. And then what we did is, once the animal is exposed, we put them for mating. The male rats will be put with female rats and look for all kinds of, see the number of pups that are generated. Okay. So there's some data I am presenting here just to give you a brief introduction. As you can see here, the first one, relative organ weights. The organ weights change, right? So in the EP1, whatever is shown in yellow, right? You see that there is an increase in organ. Increase in organ weight means there is more cell proliferation. More cell proliferation means every chance of having carcinogenic effects. Okay, so that's one effect we have seen. This is very important. You look here, I am going to explain it very slowly. F0 means whatever rats we have treated. F1 means the number of pups that are born to the treated rats. F2 means the number of pups that are born to the first generation rats. Okay. So if you look at here, in the control group means we don't treat those animals. The number of pups born to those animals are about 12, right? But if you expose these animals to low dose or high dose, you can see the number of pups have decreased to 9.5. So when you are exposed to these chemicals, expect that your fertility will decrease. Definitely. Why fertility is on the decline is one of the reasons why their fertility is on decline. And F1 you look at. F1 means the pups that were born to the FO. When they were put for mating, they also had less number of pups. which means if this generation is exposed to pesticides, not only you will have decrease in fertility, your children also will have decrease in fertility, even though they are not exposed. Got the point? This is what we call as the transgenerational toxicity. It's called as the transgenerational toxicity. So like that we have seen that up to F2 generation, even though animals are not exposed, if the grandfather is exposed to pesticides, the son also will have decrease in fertility and the grandson also will have decrease in fertility. Right? Earlier, our grandfather, great-grandfather, they used to have a lot of children. Nowadays, of course, for different reasons, we have less number of children. But if you look at the fertility aspects means the sperm count or the sperm capacity, it has come down. The reason why we are consuming a lot of pesticides, right? Of course, that's what I was telling here. The sperm count in these animals, if you look at the control, it is 105. If you expose these animals to these pesticides for 9 months, that's what is shown in green, it comes down, right? And even 12 months also it comes down from 129 to 81. And in 15 months also it comes down to 143 to 81. So it's a very, very straightforward study where doses were used exactly equivalent to humans, right? And you can see the decline in the sperm count. Of course, we have also checked the activities of enzymes which are important for sperm maturation. That is beta hydroxy steroid dehydrogenase. So their activities were also decreased in both the studies. How many of you have physiology? I hope all of you have physiology, report to physiology, right? Okay, so I will talk a little bit about sperm capacitation. What is sperm capacitation? For the sperm to fertilize an egg, it has to undergo a process known as the capacitation. That's very, very important. Okay, so when sperm undergoes capacitation, what happens is there are a lot of proteins that are removed. We use specific dyes to see the amount of protein that is present. Okay, right. So this just overview, right? As I am showing here, the sperm capacitation also decreases. Okay, this we use basing on a certain dye. The sperm capacitation also decreases. And we also looked at the hormonal profile. Hormones are very important for normal physiology, right? So if the animals are exposed to pyrethroids or if humans are exposed to pyrethroids, what happens is there are a lot of changes in the hormones. As you can see here, the yellow indicates decrease, especially GnRH. What is GnRH? Gonadotropin releasing hormone. Very important hormone, the master hormone of all the endocrine system. If that is affected, you are gone, right? So that also decreases, and luteinizing hormone, it goes up. Testosterone also goes up, it comes down. So a lot of changes in the hormonal profile if animals are treated with very low doses of the pyrethroids. Then, how many of you heard about oxidative stress? We looked at there's something known as oxidative stress in the body. If you are exposed to a lot of chemicals or any other compounds that are not good for health, there's increase in the oxidative stress. Oxidative stress leads to a lot of damages to the cell as well as to the high proliferation. And there are enzymes known as the antioxidant enzymes. Have you heard about it? Right? Antioxidant enzymes protect the body from the oxidative stress. So what happens if you expose these animals with these pyrethroids? There are a lot of changes in the enzyme activity, like the catalase, the glutathione peroxidase. These enzymes they should be functioning normal, but their enzyme activity comes down. When the enzyme activity comes down, your oxidative stress goes up and your body can be subject to a lot of damage. So we have tested glutathione S transferase, and we looked at lipid peroxidation. Lipid peroxidation also increases in these animals, right? And nitric oxide is another molecule that is very important for protecting the body from the oxidative stress. Okay, right. So, and again, antioxidant status in the EP2 protocol, I will not show this. Yeah, this is histopathological changes. What happens to the tissues when you are exposed to the pyrethroids? You can see here, when you expose animals for 9 months or 12 months or 15 months, there are a lot of degeneration of the tissues. I am not going to read all of them, but just to show you that exposure to the pyrethroids, pesticides for a longer durations will damage your tissues. We look only at the reproductive organs like the testis, epididymis and the prostate. We see a lot of changes. We also have seen that these tissues are prone to carcinoma. Okay? Right. Again. So from this part of the study, what we concluded was, this is the first study to determine the effects of a mixture of pyrethroids, and there is increase in organ weight and decrease in relative weights of some other organs. There is a transgenerational change. Transgenerational means if this generation is going to take the pesticides, that effectively is seen for the next and next and next generations. So okay, this is a very important message, that consuming pesticides is not only going to affect you, it is also going to affect the next generations. Decreased litter size and enzymes which are important for oxidative stress were also decreased. Right? Right. So the next one was, we looked at the, what is steroidogenesis? Process by which steroid enzymes are produced. It's a lot of complex process, right? There are a lot of enzymes involved and a lot of genes that are important. So I am just going to browse through this data, and here we did a PCR analysis, polymerase chain reaction. I don't know. I am sure you will have in your practicals, right? So we look at the expression of a lot of genes. What happens to the genes that are important for production of hormones when they are exposed to these pyrethroids? There are a lot of changes. Okay, so whatever is shown in the green, they are all increase or decrease. So there are a lot of change in the gene expression, right? So again, the luteinizing hormone receptor gene expression was altered, right? So then there are also something known as a sperm maturation. There are a lot of genes that are involved there, and we also looked at the factors that are important for spermatogenesis. You can see the number of genes here that are, whatever is shown in the colored title there, those genes were affected. So which means that if you expose animals to pyrethroids, it not only affects at the physiology level, it also affects at the gene level. So it's going to cause genotoxicity. It's what you call as the xenotoxicity. And of course, in the different organs we have shown them. And there are also other changes known as the epigenetic changes. What are epigenetic changes? Epigenetic means above the genome, right? So these are the changes which are very, very important for expression of the genes or for the down regulation of the genes or whatsoever. So epigenetic changes are also there, and we looked at the enzymes that are important for epigenetic changes, are the factors. As you can see here, whatever is shown in the green, those are the genes which have been altered. It's not the expression of some genes, but also epigenetic changes are also seen in animals which are exposed to pyrethroids. So again, different organs were checked, and what we did was, then we looked at the change. We have seen changes at the physiological level, like the reproductive capability, the enzyme levels. Then we went to gene level. Then we went to proteome level. Proteome means looking at the components of the proteins, right? So we did mass spectroscopy. I am not going to tell you what is, I am going to explain it. What we do is, we take control animals, animals which are not exposed, animals which are exposed. We take out the spermatozoa from those animals and isolate the proteins and subject them to sequencing, right? So then we compare our levels of these proteins between unexposed animals and the exposed animals. Okay, so I am showing you here a list of proteins. As you can see here, ratio T by C. Ratio T by C means treated by control. So all of these proteins, what happened was, though they were there in the control animals, these proteins were absent in the treated animals. You can see how many proteins have disappeared. These are all the proteins which have disappeared from the treated animals. You can see a number of proteins that have totally disappeared from the treated animals. There are so many proteins, we have shown it. Of course, some proteins have come down. Whatever, if the ratio is less than 0.5, we think that the protein has come down. then if the ratio is above 1, we think that the proteins have gone up. So there are some proteins, many of the proteins have disappeared and some of the proteins have been overexpressed. So overall what we found was there are 152 proteins which are completely absent. It's really alarming. These are male rats. They were exposed to these chemicals, and 154 proteins are totally disappeared from the spermatozoa. You can imagine the quality of the spermatozoa. So 40 were less expressed and 10 were increased. Again, here what I am trying to show you here is just focus on the colored ones. If a protein is decreased in the exposed animals, in the next generation, the same protein was found to be decreased, even though the animal was not exposed to the compound. You got the point? Right? So this very dangerous trend. Not only the exposed individuals, you look at the effects at the different levels. Physiology, there was a problem with the next generations. Gene expression also there was problem. Antioxidant enzyme, there was a problem. And even the protein levels in the different tissues were also. Oh, it's full screen. It's not full screen. So if you look at there, if a protein is disappearing in the exposed animals, the same protein is also disappearing in the pups that are born to the exposed animals, even though these new pups were not treated with the pyrethroids. It's a very alarming and dangerous trends what we are seeing here. Okay, not only one generation, F2 and F3 generations. In 3 generations, we have seen that the protein that is lost because of exposure to pesticides is again lost in the next generations. So one should be extra careful when we are talking about this one. So from this, what we concluded was there are change in the gene expression and there are also change in the protein profile in the spermatozoa of exposed rats. Right. And then when we went to the very general physiology, again the animals were exposed to two types, as I said, EP1 and EP2 means exposure protocol 1, exposure protocol 2. You can change the relative organ weights. There's a lot of change in relative organ weights. Now, till now we talked about the effects on the reproductive system. These pyrethroids are so dangerous. They're so dangerous. They not only affect the reproductive system, they affect the other organs such as the liver and the kidney. We have done liver function test and kidney function test and also lipid profile in these animals. You look at the changes. SGPT is a very common enzyme. Whenever you go to a clinic, they do that. SGOT, ALP, alkaline phosphatase, all these enzymes were upregulated. I mean, their activity was more. If their activity is more, it means that there is a tissue damage in these animals. Very dangerous trend in the liver. Of course, kidney was not any affected. You don't see any changes there. But you see changes in the lipid profile. There is an increase in cholesterol. This is very surprising. If I am exposed to pyrethroids or any pesticides for a long time, people have increase in the cholesterol levels as well as in the other lipid profile. And see the relation. Nobody will expect this. People think that I will get more cholesterol if I eat more sweets and other things. But that's not the case. Exposure to these chemicals is going to change your lipid profile. If the cholesterol increase, what happens? Your arteries will get blocked and you will get heart attack. So exposure to these pyrethroids not only causes reproductive effect, it also causes liver function test, liver function damage. It also causes increase in the lipid profile, which is very, very dangerous. These are very surprising studies. People never imagined that this will happen. Always people think that liver will be damaged because if they take more alcohol, kidney will be damaged if you take some other chemicals. But these chemicals which we are eating every day, regular basis, are causing so many harmful effects. Right? I think I am almost there, of course. And this is the liver and kidney function, the other protocol. Lipid profile and complete blood picture. We have analyzed all the parameters of the blood in these animals. Okay. So one of the interesting we saw was platelet count. the platelet count was decreasing. It increases first and then decreases. You know, platelet count is very, very important for normal physiology. And the platelet count, this was which we never expected this will happen. Exposure to these chemicals really causes such dangerous effects that it affects the platelet count. What are platelets are very important cells in the blood, right? So those cells numbers are also decreased. And antioxidant status, we have also checked in the liver and kidney. Everywhere there is a problem. Glutathione peroxidase, catalase, and glutathione S transferase, lipid peroxidation, everything was totally different in the liver and the kidney. I am just browsing this data, just to give an idea. I don't want you to go into the details of each one of these, but just to give an idea. Again, histopathological changes in these animals, in the liver, lung, and the kidney. These chemicals affect each and every part of the body: liver, lung, kidney, testes, epididymis, prostate, vas deferens, all the organs. And we have seen a lot of changes in the histopathology, which means that the organs are getting damaged. And from this we conclude that these chemicals not only cause transgenerational effects, they also cause damage to the reproductive system, and they also cause damage to the other organs of the body. You know, there is a very interesting study which came up very recently. People who spray pesticides very often develop diabetes. Diabetes we generally think is because of genetic reasons. If father has diabetes, children will also, or mother has diabetes, children also. But people who never had a history of diabetes develop diabetes if they are spraying the chemicals for these pesticides for a long time. This is very outstanding study that was done in Tamil Nadu, where they analyzed the people who are spraying these chemicals. So they saw that there is increase in diabetes. Suddenly there is increase in diabetes in particular villages. So they went and checked, and this was one of the main reasons why there is an increase in diabetes. Okay, I think this is my last slide. So overall conclusion, I would say long-term exposure results in reproductive toxicity. It causes transgeneration effects both at the transcriptome as well as the proteome levels, right? And this is a very serious concern. This study results were we sent it to the government of India. They were very, very happy to see these results that the pyrethroids are present in the food, and these are causing a lot of damages. And this was picked up as one of the best article by Current Science. And we recommended to government of India, see to it that the usage of pesticides is reduced as much as possible and increase or encourage organic production. That's what is the overall benefit or the message we give to the general population. But what I am trying to tell you is, try to avoid any food that contains these pesticides, right? But it's very difficult to avoid. But I would suggest, try to grow your own vegetables in your gardens, right? Avoid, I'm sure gardens you can without pesticides. You can have try to avoid this beverages like Pepsi, Coke, or the regular bottled water, right? Because of course older generations, they are gone, but younger generations, what happens is, if you consume pesticides on a regular basis, not only you are affected, your next generations are also affected by a lot of changes. This is the one something message I wanted to tell you, right? I hope I was able to catch your attention. There's a lot of research study here, but I had another presentation which was very general, how pesticides act on the nervous system and all that. I thought that's a very regular textbook topic, but this I thought let me convey you some important message. You spread the message, not only to you, this message is for everyone. Avoid using these chemicals as much as possible, right? Thank you so much. Madam, is it okay? Right. So I hope I did not bore you for a long time. If there is anything you want to ask, you can ask. And before we talk, I want to tell you some general things. I am from University of Hyderabad, School of Life Sciences, Department of Animal Biology. Okay, our School of Life Sciences, I have to tell this to you, has 65 faculty members, Madam knows all the history. 65 faculty members currently. We have a huge building, brand new building, and different 5 departments: animal biology, plant biology, biochemistry, biotechnology, and bioinformatics. All faculty are excellent. They are excellent. Nobody is taken, in very rare cases, majority have taken after a postdoctoral fellowship in the USA or some other places. They are highly, research wise as well as knowledge wise, they are very good. And in School of Life Sciences, we have a small unit known as the Skill Development Center. We have started it in 2019. Skill Development Center was started because there is a saying, very common saying in our country: all our students are educated but not employable. Employable means you all will have degree after 2 years or 3 years, but when you go to the job market, you are not taken. What is the reason? Because you lack the skills. Whenever you go for a job, nobody looks at your degrees or marks. The only thing they look at is your skills. What skills you have, so that I can give you this job. So that is the reason why we started this Skill Development Center in 2019. We train students in a lot of subjects. Recently, the government of India gave 85 lakh rupees to train students from rural areas. Pick up students from rural areas means whoever is studying in colleges of rural areas. Pick up. We, of course, throughout India we advertise, and then we get about 200 applications, but the limit is 30. We have to take only 30 students, following Government of India reservation policy. We have picked up last October, November, we have done one training program. It is for 6 weeks. So we start from preparation of a solution all the way up to doing CRISPR-Cas9 gene editing. The students are trained in all this, hands-on. Only 30. There are about 14-15 faculty members who come forward and then impart the training. We provide them free accommodation, we provide them free food, and we waive off all the training fees. Right? All the fees are removed. So which means that we invest at least 50,000 rupees on each student. So like that we get 30 students, including the cost of accommodation, food and everything, around 50 to 60,000 we invest on each and every student. So I would suggest you to apply. The eligibility is any student of biology, both graduate as well as postgraduate, can apply for this program. We advertise it. Okay, you please apply, and if your luck is good, you will be selected. The condition is you should have a CGPA of minimum of 8. 8 should be the CGPA. Then if you have 8, then of course we don't take more than 2 to 3 students from one college. So you please apply to this program. Madam? Passed out students are also eligible. Yes, madam. Most of the students who are trained in our skill development, they either go for a PhD in India or abroad, or they go for a job. Okay. So you please apply. I will suggest all of you to apply for it. And beyond this, we also conduct other programs which are all charged. We conduct summer internship program which is for 2 months. We charge 30,000 rupees, and hostel and food are extra. We get around 60 students per year. So 60 into 30 is 18 lakhs. Some amount of the money is taken to train students from rural areas. Some amount is set aside. We also have something called a long-term internship program. They come for 6 months. We charge 60,000 rupees. Right? So, and we get around, there's a limit, we take only 20 of them. And when students do this summer internship or long-term internship, we have seen the trend that they get admissions in very good institutions. So they are trained thoroughly, and they get very good institutions. Of course, there are some cases where we give fee waiver, but not very regularly. And we also conduct small programs, 1 week, 2 week programs. So keep looking at the Skill Development Center, School of Life Sciences, University of Hyderabad. You will have a lot of opportunities there. Okay. There is something known as the student observership. Student observer means you will come to our labs, you will be there for 15 days or 1 month depending on the college. I think I discussed with Anusha madam also. The student observership, hopefully we will be able to conduct for you. We talked to madam and I agreed that I will host 20 students. The only problem we have is the accommodation. Our hostels are so full, the guest houses are so full, getting accommodation has become very difficult. But let me see. But for 20 students, for this year, madam can recommend any 20 students. You can come there for 2 weeks, maybe head of the department and madam can sit together and select the students. It's your decision how to select and send them. They can be there for 2 weeks. If you look at the building, if you look at the research lab, then you get a lot of encouragement. Oh, I also want to do this. Okay, so that part we can take care of. Okay, please talk to your head of the department and madam. Okay, right. Madam, any comments?
The session is open for discussion. If students, if you have any kind of doubt regarding internships or whatever sir has talked upon subject wise, you may ask.
Any question is fine. Career related also is fine. If you have any doubts on the career, no problem. You can ask.
Life sciences related, any question?
Sir, is there any potential replacement for pyrethroids as of now?
Natural compounds.
Okay.
Synthetic compounds are very dangerous, but natural compounds, they're very costly to produce.
Yeah, I don't know. You are talking about even purification of water also,
Yes, sir. For purification of water,
at least.
it is used, yes, sir. But nowadays people say that no, you can use ozone.
Okay.
Ozone is one compound which they are used, but it's not as effective as the pyrethroids. Pyrethroids are more commonly used. There is no replacement, but people are coming up with natural molecules in the form of nanoparticles to spray on the crops.
Okay.
And they are also coming up something as cry proteins. You heard about it? Plant biology people, they do. These are cry proteins which inhibit the growth of the insect.
Okay.
So
They
natural
directly
compounds are being developed, but it's not as fast as they develop the synthetic compounds. Right.
Thank you, sir.
Anyone else?
Students, you should ask some questions, or else I will be disappointed. No, madam. Some questions should come up. Any question? Okay, anybody who wants to go to abroad for higher
Yes.
education? You want some?
I mean, in the paper, sir, you're emphasizing on the reproductive system. Is there any reason, like, specially affecting the reproductive system? Why is it specially affecting the reproductive system?
It's not only affecting the reproductive system, that's one of the parameters we checked. But as I also shown you that it affects the liver and lung and the kidney also. But our interest is reproductive system because we are interested in the generations. Whether the main reason why we analyzed was whether it affects next generations. So that's why we focus on reproductive system. Okay. Any other question, madam? Okay, I have a question for you.
It's good, sir.
How much is 25 by 5?
25 by 5.
25 divided by 5. Really? Think about it. 25 divided by 5. I can't write. If I can write, it will be very nice. 25 divided by 5. Okay, I will tell you the answer. You are putting 25 divided by 5. Right? That's what you are writing. Let's go back to our school days. 25 left 5. Isn't it? That's how you used to do in school days. I used to do that. 5 bracket 25 bracket. Remember it? Okay, now 5, I put 1. I put 5 here. Subtract 20. I put 4. 20, 0. Got the point?
But you are coming from right side, sir. It should be from left side.
Madam, if that is taught to you from your childhood, you would have followed it or not?
Yes, sir.
So 25 by 5 can be 5 or 25 by 5 can be 14. So think in that direction. Then you will excel. Right? So students should think in the other way. This is what we call as out of the box thinking. Right? Don't always think, see, I don't know. What is the color of that fan? Brown.
It's not white.
It's not,
Maybe.
it's not white, but if you have been told from childhood that is green, you would have told green. So most of us depend on knowledge which is generally told to us. Try to think beyond that. That's what I want to tell you. Okay? If somebody has said that is red from your childhood, you would have said, if I asked what is the color, you would have said red. Why can't it be red? Right? Same way 25 by 5 need not be 5. 25 by 5 can be 14. This is what we call as there are two types of developments. One is sequential development. There is something as a disruptive development. Disruptive development means, one of the best example is, nobody imagined that one day we will drink water only from bottles. Earlier we used to drink water from taps when I was a child. But now, wherever you are there, you drink water from bottles. So this Bisleri is the first company which came up with that idea. Right? Providing water in bottles. That's what they call as a disruptive thinking. Same way cell phones. You can imagine the days when we used to use a regular phone. Cell phones are the disruptive thinking. Right? So try to think in a disruptive way. Disruptive way means not to damage it, but in a way that is multiplied many times. That only you can do it. Right? Madam, any other questions?
Any other questions?
That boy has one question. No, he wants to register. Okay. No problem. He's more interested in attendance than question.
Sir, good afternoon.
Good afternoon, madam.
ఎలాగో మనం పెస్టిసైడ్స్కి వాటికి అలవాటు పడిపోయాం కాబట్టి క్యూర్ ఏమై ఉంటుంది సార్ అదర్ దెన్ ప్రివెన్షన్?
Prevention is better than cure. Nothing. There's nothing. No way you can.
బట్ మన బాడీలో ఆల్రెడీ అది నార్మల్గా
Yeah, if you stop taking, you will be fine after some. This is what we call as the reversal effects. You give the chemical to the animal, look at the effects, and later on you withdraw it. Then the physiology comes back. మళ్ళీ మామూలు అయిపోతుంది అది. ఓకే, మళ్ళీ మానేసాం అనుకోండి. ఇప్పుడు ఫర్ ఎగ్జాంపుల్ స్మోకింగ్, ఆల్కహాల్ ఉంటాయి, దానివల్ల చాలా డ్యామేజెస్ ఉంటాయి. కానీ వాళ్ళు మానేసిన తర్వాత దే బికమ్ నార్మల్. సేమ్ వే స్మోకింగ్, ఆల్కహాల్ అలవాట్లు అంటాము. ఇవి ఏమంటారు దాన్ని, నెససరీ ఈవిల్ అంటారు. తెలుసా మీకు? యూ నో వాట్ ఈస్ నెససరీ ఈవిల్? యూ కెనాట్ అవాయిడ్ ఇట్. ఈ పెస్టిసైడ్స్ మనం తిని తీరాల్సిందే, వేరే దారి లేదు. మీకు వద్దు అనుకుంటే యూ గో ఫర్ ఆర్గానిక్ ఫుడ్స్, విచ్ మీన్స్ దట్ యూ గ్రో ఫుడ్. మీ ఇంట్లోనే పెంచుకోండి మొక్కలు, కూరగాయలు, ఇవన్నీ.
బట్ ప్రొడక్షన్ కోసం ఖచ్చితంగా వాడాలి కదా?
ఖచ్చితంగా వాడాలి. ఏం చేస్తాం ఇప్పుడు? పాపులేషన్ పెరిగిపోతుంది, జనాభా పెరిగిపోతుంది, ఫుడ్ కావాలి. ఫుడ్ కావాలంటే స్ప్రే చేసి తీరాలి, తప్పదు. అందుకని ఏం చేయాలి? ఆర్గానిక్ వెళ్ళాలి, లేదు పాపులేషన్ తగ్గించుకోవాలి. అది కాని పని అది. రైట్? పాపులేషన్ తగ్గించి అంత ఈజీ పని కాదు. సో ఇది మనం ఏం చేయలేము. కాకపోతే ట్రై టు బీ వెరీ కేర్ఫుల్. అది ఓకే. రైట్. Any other question?
Sir.
Good afternoon, sir.
Good afternoon, madam.
Is microbial pesticides or biofertilizers? Biopesticides?
నాకు అర్థం కాలం.
Is microbial pesticides or biopesticides, sir?
Yes, madam. Microbial pesticides are also there. There are some microbes. They take it in nano formulations. అవి
How
స్ప్రే
can be
చేస్తారు.
produced by naturally, sir?
So there are some microbial organisms which can specifically target fungus. They are used for fungal infections. So bacteria.
Thank
Yes.
you, sir.
So they are used. Bacteria are generally used as biopesticides. Right?
Thanks.
Okay. One last question we will close. Right? What is the age of humans? What is the lifetime of humans? Lifespan? 100 years. That's why we ఏమంటాము శతాయుష్మాన్ భవ అంటాం. అవునా? సో 100 సంవత్సరాలు బతకమంటాం అందరినీ. ఈ 100 సంవత్సరాలు ఎలా వచ్చింది మనకు? డిడ్ యూ ఎవర్ థింక్ ఈ 100 సంవత్సరాలు మనకు ఎలా వచ్చింది? దీన్ని మీకు ఒక ఎగ్జాంపుల్ చెప్తాను. ఈ సృష్టిని ఎవరో సృష్టించారు. అవునా? ఎవరన్నా కావచ్చులేండి. సృష్టించిన తర్వాత మహానుభావుడు ఏం చేశాడంటే అందరికీ ఇది ఇవ్వాలి, లైఫ్ టైం ఇవ్వాలి కదా. బ్యాక్టీరియా అయితే 20 మినిట్స్, అదే ఒక కుక్కకైతే 20 సంవత్సరాలు అలా ఇవ్వాలి కదా. సో ఆయన ఏం చేశాడంటే ఇలాగే ఒక కాన్ఫరెన్స్ పెట్టాడు. ఎవరు? ఆ సృష్టికర్త కాన్ఫరెన్స్ పెట్టి ఆయన ఒక లెక్క వేశాడు. ఎన్ని జీవరాశులు ఉన్నాయి, ఈ ప్రపంచం ఎన్ని సంవత్సరాలు ఉంటుంది, దానంతా బేస్ చేసుకొని వచ్చిన వాళ్ళందరికీ నీకింత 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 అని ఇచ్చేసాడు ఆయన. ఈ మీటింగ్ పెట్టాడు, అందరూ ఒప్పుకున్నారు. బ్యాక్టీరియాకి 20 నిమిషాలు, ఓకే, ఏనుగుకి 100 సంవత్సరాలు ఏదో ఇచ్చాడు. ఈ మీటింగ్కి 4 రాలేదు. 4 జీవరాశులు రాలేదు. ఒకటి న్యాచురల్గా మానవుడు, అహంభావుడు రాలేదు. రెండవది గాడిద, దానికి ఏం పోతాంలే అని అది కూడా రాలేదు. మూడవది కుక్క. నాలుగవది ఇది గుడ్ల గుబ్బ అంటారు, ఆవులు తెలుసా? ఈ 4 జీవరాశులు రాలేదు. సరే లేట్గా వచ్చినా ఈయన ఇంకా కాన్ఫరెన్స్ అయిపోయింది కదా, బయలుదేరదాం భోజనం చేద్దాం అనుకుంటే వచ్చాయి ఈ 4. స్వామి లేట్ అయిపోయింది, కొంచెం మాకు కూడా ఏదైనా లైఫ్ టైం ఇస్తారా అంటే ఆయన బుక్ అంతా చూసి 160 సంవత్సరాలు మిగిలి ఉన్నాయి, ఒక్కొక్క 40 తీసుకోండి అన్నాడు. 4 ఉన్నారు కదా, ఓకే నువ్వు 40 మీ లైఫ్ టైం ఇది అని చెప్పేశాడు ఆయన. సరే హ్యాపీగా గాడిద, కుక్క, గుడ్లగోబ సంతోషంగా ఉన్నాయి. మానవుడు తెలుసు కదా అహంకారి, మళ్ళీ కుక్కకి 40 సంవత్సరాలు, నాకు 40 సంవత్సరాలు, మేమిద్దరం ఒకటే బతికేది ఏంది అని అడిగాడు ఆయన. గాడిదకి 40 సంవత్సరాలు, పనికిరాని ఈ పక్షికి ఊరికే చెట్టు మీద కూర్చుంటుంది దానికి 40 సంవత్సరాలు, నేను మహామేధావి, నేను నాకు 40 అయింది, వాళ్ళకి 40 అయింది అన్నాడు. ఇంక ఇది గొడవ జరుగుతూనే ఉంటే ఇంకా లాస్ట్కి గాడిద ఏం చేసిందంటే, నా జీవితంలో నేను ఏం చేస్తాను, బరువులు మోసేదే కదా, 40 సంవత్సరాలు ఎందుకు, నాకు 20 సార్లు అనిందంట. సరే ఏం చేసినాడు ఆయన, ఓకే గాడిద 20 ఇచ్చింది కదా తీసుకో అన్నాడు. సో ఆయనకి ఎంత అయింది ఇప్పుడు? 60 అయింది. అయినా ఒప్పుకోలేదు. కుక్కకి 40 అయింది, నాకు 60 అయింది, 1:10 రేషియోనే ఉండాలా, ఏదో ఆర్గ్యుమెంట్ పెడతాను. ఇంక చివరి కుక్క కూడా ఏమైందంటే, నేనేం చేస్తానండి జీవితకాలం కాపలాగా చెప్పను కదా, సో ఇంకో 20 ఇస్తానని చెప్పింది. ఎంత వచ్చింది ఆయనకి? 80 వచ్చింది. అయినా ఒప్పుకోలేను. 80, 40, గుడ్ల గోబకి 40 అయింది, నాకు 80 అయింది, 1:2 ఇది కుదరదని మళ్ళీ ఆర్గ్యుమెంట్ పెట్టాడు. ఇంకా అది జరిగి ఆమె కూడా ఆ గుడ్ల గోబ కూడా 20 ఇచ్చేసింది. ఎంత వచ్చింది ఇప్పుడు? 100. అలా వచ్చింది. ఇక్కడ 100 ఎలా వచ్చింది ఇంపార్టెంట్ కాదు. ఫస్ట్ 40 years ఆయన ఒరిజినల్ లైఫ్, 40 to 60 గాడిద జీవితం, 40 దాటిన తర్వాత పెళ్ళి బిడ్డలను ఎత్తుకొని ఈ భారాలు మోస్తూ బతుకుతాం 60 వరకు. అవునా? 60 నుంచి 60కి రిటైర్ అవుతాం. 60 నుంచి 80కి ఏం చేస్తాం? ఇంటి ముందర కూర్చొని వచ్చేపోయే వాడిని మీద అరుస్తూ ఉంటాం. అది కుక్క జీవితం. ఇంక 80 అయిన తర్వాత చేసేదేముంది? ఒక మూలలో కూర్చొని ఉంటాం అంతే. అది గుడ్లగొవ్వ జీవితం. సో మనం ఎన్ని సంవత్సరాలు బతికినా మన జీవితం ఎంత? 40. ఏం చేయాలన్నా 40 లోపల చేయండి, ఆ తర్వాత చేయలేరు.
I thank all the listeners here, including UG students, PG students, research scholars, lecturers, and I thank Professor Y. Suresh. So, take home message, apart from the experiments and the findings which sir had made, the take home message is you should not use chemicals indiscriminately. Especially our area is infested with mosquitoes, so unavoidably we tend to use mosquito repellents. So you have to remember that it is going to affect the reproductive system, especially in the males. And also we should stop using perfumes. As sir has told, some of the sprays, including the perfumes, are inducing diabetes epigenetically. Epigenetically means you are not genetically inheriting it. It is not in your parents, but it is found in you. Diabetes is coming to the generations where it is not found in higher generations, right? So these two take home messages you should remember, and try not to use mosquito repellents and sprays. Yes? Now I invite Ms. Parnika Sai to give vote of thanks for this session, for this day.
A graceful noon to all the dignitaries and each and everyone in this event. It's my privilege to propose a vote of thanks and acknowledge the contribution of those who worked really hard to make this popular lecture series ABC 2024 happen. On behalf of our biotechnology department, I extend a really hearty vote of thanks to each and everyone. An event like this cannot happen in overnight. The wheels start rolling week ago. It requires planning bird's eye for details. We have been fortunate enough to be backed by a team of very proactive and dedicated principals who is well versed in his job. I am in short of words for his involvement and willingness to take on the completion of tasks beyond the comfort zones. So first of all, I am expressing our sincere thanks to our young and dynamic principal sir for being the catalyst that stimulated us to do our best and standing as pillar of strength with a deep sense of appreciation. I take my immense pleasure in extending sincere thanks for guest of honor C.H. Krishna sir, RJD CCE Zone 2, who had highlighted the directions for taking the talk to greater heights. My heart goes out to thank our respected chief guest Dr. P. Vijay Nirmala ma'am for accepting our invitation and coming to this ABC 2024 and encouraging the morale of the speaker by her gusto in front-end biotechnology. I also wish to have special recognition for the resource person, that is today's speaker, Dr. Professor Y. Suresh, Department of Animal Biology, University of Hyderabad, who delivered a fruitful talk on the topic effect of pyrethroids at doses relevant to human consumption on the general and male reproductive physiology in rats by giving insightful views on how much dose people are exposed to. By sparing his time from his busiest schedule to grace the occasion in an extraordinary way. We thank you, sir. We are all happy that this Department of Biotechnology Conference, Travel, Exhibition, Popular Lectures, that is CTEP, has been granted popular lecture series and conducted extremely well. And our sincere gratitude goes to all the organizing committee members for your rock-solid support system and encouragement. A special mention to Lakshmi Narayana sir, Murthy sir for this special ambience, and System Administrator Praveen sir for live streaming of YouTube. Finally, I would like to thank our budding rock stars of biotechnology, that is biotechnology students and student committees. We also offer a special thanks to non-teaching staff, and we thank all the participants for being a part of this popular lecture series, Advances in Biotechnological Chronicles 2024. Thank you.